All right, I think we'll go ahead and get started. I anticipate more will join as things move along, but in the interest of time, we want to make sure we cover everything we aim to share. So we'll get started. Thank you all so much for joining today. I'm Jen Tozer. I'm the Executive Director of a Foundation Building Strength. I'll be helping facilitate this research update and information session from a Foundation Building Strength, along with 𝗏𝗏𝘅 𝗔 𝗙𝘂𝘂𝗶𝗴𝗵. I do practice saying his last name, which is amazing last name. He's AFBS's scientific director, and he and I will start out the session with sharing a bit about where we are in our research progress, and how you all can contribute to the advancing research towards finding treatments for Nemaline myopathy. A few things I want to note before we get started. This session is being recorded. It will be shared afterwards. We will have a Q&A for Gus and I's portion of the session at the end after we hear from Dr. Gupta and Dr. Dowling. So just hold on to your questions, and we do want to get to them, but we also want to make sure we give plenty of time for the research update portion. But we do encourage questions, so please put them in the chat, or you raise your hand, and we want to address as many questions as possible. So today we will be, like I said, first covering the AFBS's investments and the role you can play as a community member in advancing our research. We also want to share a little bit about the development pipeline in terms of where we are in the pipeline towards finding treatments for NM. So Gus will be sharing that, which I think will be really helpful. And then we'll hear from Dr. Dowling on his research. That will be followed by a Q&A, and then we'll hear from Dr. Gupta on her latest research developments, followed by a Q&A. And then, as I said, at the end, we'll address any questions about anything related to AFBS's research beyond Dr. Gupta and Dr. Dowling's work. And that is just to ensure we have plenty of time to our researchers so we can hear from them. And then if we need to go beyond the 90 minutes that we've scheduled for this session, we can do that with no problem. All right, well, I'll start with sharing a little bit about where we are in our investments and moving the needle towards finding treatments for NM. We're very, very excited and proud to share that we have agreed and committed to investing $3.6 million over the next 3 years, which is an incredible investment, and it's thanks to our individual donors who are supporting our work. We're actively invested in supporting 9 research projects right now around the world, and researching 6 genetic subtypes. As you can see on this slide, we're investing in research infrastructure and research to develop gene therapies. They're both equally important and critical to getting us to get treatments to our community. So in terms of our infrastructure, we're really thrilled to share, if you haven't seen already through our emails and social media, that our natural history study in North America is officially live and recruiting. There are at Stanford, and there will be other sites that will be recruiting to support this work very soon. We have a lot of information about this. I'm not going to go into too much detail, but this is a really critical piece of the puzzle to move things ahead. And we also have a natural history study in Brazil. For any of those watching this that are in Brazil, you can participate in the natural history study there. There is also an affiliated UK study that AFBS is not funding, but there is collaboration there. Also, we're funding the biobank, the NM biobank, and genetic testing at the Beggs lab at Boston Children's Hospital, which is another piece that I'll briefly cover. And then on the research pipeline side, the research projects, I have a list here of all of the research projects that we're supporting. we're so thrilled that we'll be hearing from Dr. Dowling and Dr. Gupta today. And we also have additional projects listed here from Dr. Ottenheim, Dr. 𝗏 at Nationwide Children's Hospital, Dr. Mack at University of Washington, who's partnering with Dr. Howling. And then we also have a very exciting new partnership that's with DNA Nanobots, which is our first industry partnership. While I won't go into all the details, Gus will share a little bit more about the research. You can see which research projects we're actively invested in and also the genes that we're diversified in supporting. I do think it's really important for our community, for the NM community, to know that supporting research is not just investing in the research projects, but it's also participating in patient registries, in the natural history study, and helping us get ready for clinical trials. This is essential. Without this involvement, we won't be able to bring these potential therapies to the community. And of course, fundraising and awareness is a critical piece. We just had a very successful NM Awareness Month in May, and that really helped bring more awareness to the disease, which is very important to our progress. And also fundraising. AFBS is exclusively funded by individual donors, so every single donation matters. We don't have federal funding. We really don't have corporate funding or other grants. We're getting every one of our dollars from individuals. So you all and our donors are making this progress happen. So thank you for this. I want to get into just a few things to consider that you can do as an affected individual to support this work. But of course, if you ever have any questions, please reach out to me or any of the AFBS team, and we would be glad to assist with any questions, anything that you're interested in getting involved in. A few things that I want to highlight here, and I am going through this pretty quickly, but I encourage you to follow up with questions at the end or by email, or we can set up a call to go over this in more detail. But a few key pieces, if you haven't already, please register in the CMDIR. This is a way for us to collect every individual around the world that is affected by NM, and it's really important for our researchers to understand this data and use it for advancing us into clinical trials. If you haven't already received your genetic testing to confirm your diagnosis and your genetic subtype, this is a really important piece as well. And we're happy to support you in getting that information if you haven't done it already. Stacy, who I think is on the call today, is able to assist with this. And so I put her email here, and also the 𝗏𝗃's lab can also assist with this if it's if needed. And also, in addition to what the 𝗏𝗃's lab can support in genetic testing, this is the hub of all NM tissue donations. And this is a really important piece to support research now and in the future to be able to test the therapies and develop our science using a variety of different NM genetic subtypes and tissue samples. So please consider that in a future surgery. And again, 𝗏𝗃 Cassette will also be able to assist with that and getting you more information. One piece that you may not know is something called an ICD-11 code that clinicians use in their entering information about your care. It's important that your clinician is using the correct code so we're able to access comprehensive data, and in the future researchers can also see this information. So just something to consider, telling your clinician that Nemaline myopathy has an ICD-11 code, something that is actually really important for accuracy in medical records. And also the natural history study. Like I mentioned, we're so thrilled to have the North America Natural History Study underway. There's also the Citizen Health retrospective natural history study that is still collecting patient records in order to develop a de-identified study, to do a natural history study. So particularly this prospective natural history study, or I'm sorry, retrospective natural history study, will be looking at the NEB and ACTA1 gene for all ages and US based. So we encourage everybody to participate in Citizen Health. It's a wonderful research. It has incredible AI tools to help support your care and access all of your medical records, but we also have a wonderful benefit to advance research through this retrospective natural history study. And lastly, like I said, funding is critical to all of this. We wouldn't be able to advance research without donations. So I just encourage you all to spread the word. If you're able to donate, if you're able to ask a friend or family to support AFBS, it's a really important piece of all of this. So I know I buzzed through all that pretty quickly, and you may have questions. I'm happy to put some of the links into the chat when I pass this over to Gus. But I just wanted to emphasize the importance of how you can contribute if you're not already participating. And I know many of you already are doing all of these things or many of these things that I listed. So thank you for your contribution and involvement. Now I'm going to pass it over to Gus to talk about the therapy development pipeline. Gus, you want to take it away?
Yes. Hello, everyone. And I will share with you 3 slides to show you where we are and where we are going in the pipeline of developing treatments for different types of pneumonia myopathy. So let's first discuss how does an idea in a research lab becomes a therapy. And for every approved treatment from aspirin to gene therapy, the approval travel or will have to travel this same road. It must follow an established regulatory pathway guiding the process, and AFBS accelerates the journey from lab breakthroughs to life-saving treatments by providing funding support at every stage possible. AFBS has invested in basic research or discovery research, mostly in the past. We funded research to understand what genes cause NM and how the different mutations in the different genes provoke the different types of pneumonia myopathies. So responding to questions like, what does this gene or protein do? Or why do muscle fibers break down in alpha-actinin or actin or enabling is affected? And basic research also involves the creation of tools to do these studies, creation of animal models, cell models, histochemistry, microscopy to analyze the pathology, etc. So with this fundamental knowledge, you can start generating ideas to fix the problems. For example, in prior basic research, science may have identified if the disease is dominant or recessive and design methodologies with potential to fix the issues. So you can start testing with the best tools you have available at the moment to obtain proof of concept, which means showing that an idea for a therapy has potential. And you try to make better tools to prove your hypothesis further. So that's the first part of the pipeline that you see there. Then you will use the best tools and the best therapeutic candidates to develop further, showing that you can reverse specific pathology marks and symptoms in different models of the disease and evaluating potency, efficacy, safety of the approach that you are developing. And if your candidate shows these desirable effects, you can advance to translational development. And in the translational phase, the scientists do more work to bolster the idea that the development candidate could feasibly be manufactured and administered as a therapy. They carry out preliminary safety experiments on animals. They may also experiment with optimizing the efficacious dose in the animal model and what is the best way to administer the therapy. And after gathering all this data, the scientists arrange a pre-investigational new drug meeting with the FDA here in the US, similar pathways in Europe or other countries. And at that meeting, you obtain feedback from the FDA on the remaining preclinical work they will do prior to applying for permission to carry out the clinical trial in humans. So then the late stage research, we can call it the IND enabling research in this pipeline. The investigators plan out a formal clinical protocol for a clinical trial. And all this information is submitted to the FDA in the form of an IND application. And if the FDA is satisfied with all the information provided, they will give permission for the investigators or the clinical trial sponsors to initiate a clinical trial. And clinical trials traditionally proceed in 3 phases, each of which must be reviewed by the FDA prior to advancing to the next stage. So you have heard about phase 1, phase 2, phase 3. Phase 1 is the clinical trials that include a very small number of people and are primarily intended to test whether the proposed therapy is safe, but also to look for early signs that it may be efficacious in treating the disease. And in addition, different dose levels or dosing regimes may be explored during the phase 1 clinical trial. Then if that goes well, you are approved to go to phase 2, and the clinical trial includes slightly more people and continue to monitor safety while starting to look more rigorously at signs that the therapeutic is efficacious. And then the phase 3 clinical the trials are done if there are signs that the therapy has efficacy in the phase 2 trial. The phase 3 include larger number of people and usually have a control arm in order to get statistical data supporting the idea that the therapy is truly benefiting by patients. And then it will come the phase of commercialization. After phase 3, the sponsor may apply for a license to manufacture and sell the drug. And FDA reviews all the preclinical, manufacturing, and clinical trial data, looking for evidence that patient benefits from taking the therapy and that this benefit outweigh any safety risks. And if the FDA decides the therapy is safe and effective, it may grant level approval, and then and only then the therapy can be used by doctors to treat patients. So the path is long, but AFBS funds research at multiple stages simultaneously, so progress happens in parallel. The next slide. After reviewing the development pipeline for therapies from discoveries and preclinical testing in the research labs all the way until clinical trials, let's focus on where we are and where we are going. As mentioned earlier, we have supported important discovery research, and we are currently funding proof of concept studies and development of therapeutic candidates. Then ahead, we will support working on the regulatory package necessary to file for an IND application with the idea of obtaining to get to the goal of conducting clinical trials and hopefully approval of therapies for nemaline myopathy. And the next slide, we can see that the research funded by AFBS today, where it lives today, and we are at the initial and medium-term stages of testing different therapeutic candidates and moving forward the better hits. Here we can see the stage where all the projects funded by AFBS currently are. In the proof of concept space, we have a non-viral gene delivery system for the very large gene 𝗫, as well as a very novel approach of gene therapy using a stitching system to reconstitute a large protein as 𝗫 from administration of different fragments. Because as probably you know, a large gene like 𝗫 with the current technology of AAVs it's impossible to deliver that big size of a gene. So the scientists need to be creative to find ways to insert this huge gene in the genome of the patient. Dr. Dowling will talk about this next. We also have projects in the more advanced stage of refinement of potential candidates. For example, the work from Vandana Gupta, our other guest speaker today, and work at Afro's Russian non-shared lab with a promising gene therapy system for ACTA1. Or we can also mention the work by Dr. Koen Ottenheim, developing an inhibitory RNA to silence the mutated copy of the KBTBD13, and also the creation of a very important tool, which is a human model of Nemaline myopathy by generating muscles in 3D or organoids, muscle organoids, to test the different therapeutic approaches. And finally, a substantial part of the budget for research is going to build infrastructure and capacity. As Shen mentioned earlier, for example, we are funding Nemaline myopathy natural history study in North America and also in Brazil. And we also fund the biobanking and genetic testing at Boston Children's Hospital in Beck's lab. So I hope this is helpful to you to see how AFBS and your contributions are accelerating the bench to bedside process for a discovery in the lab to a real therapy for patients. So as Jen mentioned, we can have more discussion about this at the end. But now let's move to the best part of this webinar, which is the talks by our guests. We will first have Dr. Shin Dowling, followed by Vandana Gupta, I think, or the other way around. Take the lead, you, Shen, again. Thank you all.
Thank you so much, Gus. That was very helpful to hear that, and we certainly encourage questions, like we said at the end, so hang on to those, and we'd love to address any questions you all have. I think we will shift our order a bit because Dr. Dowling is still joining our call, so we're going to have Dr. Gupta go first. I hope that's okay with you, Dr. Gupta. Thank you. So I'm just going to skip ahead to Dr. Gupta. We're so proud to be investing in Dr. Gupta's work. She's making incredible progress, and I really won't go beyond that, and I'm going to let her share the work that she's doing because it's really incredible and exciting, and she's certainly the most knowledgeable to share. So I'll pass it over to you, Dr. Gupta, to share your slides.
All right, thank you, Jen. I had to send a request to share my slide. All right. All right, everyone. It's so wonderful to be here today to share our work and nice to meet new family as well as to see so many familiar faces. We are based in Boston. My lab is at Brigham and Women's Hospital, where we are working on developing therapies for a number of neuromuscular diseases. And one of the major focus of my group is Gnumlin myopathy. Just give me 1 second. All right. So Gnumlin myopathy is a genetic disorder. The disease is caused by mutation in any of the 12 genes, Gnumlin, actin, 𝗏 genes. The good news is that the progress in research has made it possible to now have genetic diagnosis in about 70% of the Gnumlin patients. The disease is clinically heterogeneous. Most of the patients have muscle weakness. But in addition to muscle weakness, depending on the clinical severity of the disease, some patients may also have breathing and feeding difficulties. And in spite of this much progress in the diagnosis of the genes in the Gnumlin myopathy, there's still no cure and treatment options are really limited. Right now, the treatment is pretty much focuses on the care and managing disease symptoms. And even though the disease is caused by mutation in 12 genes, one of the common themes that we see is that when we look at the skeletal muscle under the microscope, we see these dark structures, which are called Gnumlin bodies. And that's why the disease has the name Gnumlin myopathy. So before we go into the therapeutic aspect of the therapy, I just want to quickly remind everybody the recessive form of the disease. So today the focus of my talk is going to be mostly on the 𝗏𝗏𝘁 mutation. 𝗏𝗏𝘁 is a recessive disorder where each of the parent is a carrier of the disease. So they have a working copy of the disease, which is shown here, working copy of the gene in the blue, and a non-working copy of the gene in the pink or purple. So these carriers, they are perfectly healthy, perfectly normal. And in rare situation, one of the kid can have both non-working copy of the disease where the gene does not make any protein, resulting in the weak muscle. So over the years, a number of different patients have been described in literature. Right now, 31 different 𝗏𝗏𝘁 mutation, which can be in any part of the gene or protein, have been described in 80 patients. But this is just the tip of the iceberg because these are the cases in which the patients have been described by the labs that go ahead and publish these results in the literature. But there are many labs that do not publish this result, and there are certain regions that are not that academically oriented, or some patients prefer not to pursue these things further. So in order to get a good understanding of the prevalence of the disease, we have looked in the databases. One of the database which is available to us is at Broad Institute. So what they have done is, for each gene that is associated with disease, they have looked at the carrier frequency in different ethnicities. So as we can see that for 𝗏𝗏𝘁, different ethnic areas or different ethnic population have different prevalence of frequency by which they can have 𝗏𝗏𝘁 mutation. And we have used this data to have a rough estimate of how many 𝗏𝗏𝘁 kids who will have both non-working copy of the gene or that are affected by the disease are born each year. So in some cases, there are very low estimate of 𝗏𝗏𝘁 mutations, but in some ethnicities like East Asian or South Asian, we see high prevalence of 𝗏𝗏𝘁 mutations. So if we combine all of this estimate from different regions, we can see there are about 40 to 45 kids who are born with 𝗏𝗏𝘁 mutations every year. And 𝗏𝗏𝘁 is a severe form of the disease. Most of the baby experience very severe disease onset at birth or within few months after the birth. So this puts a very strong need and unmet need to develop therapy for 𝗏𝗏𝘁 patient. But one of the things that emerges from the studies is that 𝗏𝗏𝘁 is not as rare as was thought few years back. I know a few years back, we only knew one family, which is here, but now within last few months, we have been contacted by multiple families with baby born this year. So when we talk about therapy for 𝗏𝗏𝘁 patients, we decided to develop a gene replacement therapy. And I'm just going to spend a couple of minutes here to go over what is a gene replacement therapy. So as I mentioned, when we have two copies of the mutation or non-working gene, the muscle does not make any protein. So in this case, we do not see any 𝗘𝗪40 protein in these patients, as a result of which the muscle is very weak. And what we have done is we have used a carrier, which is a virus called adeno-associated virus. So we use the shell of this virus and we package 𝗘𝗪40 working copy into this carrier and put it back in the muscle. So now this working copy goes in the muscle and it starts to make new 𝗘𝗪40 protein, which results in formation of new and healthy muscle. And this concept is not really new. There's already been a gene therapy for spinal muscular atrophy, which is very similar to 𝗘𝗪40 patient. Those patients also have very weak muscle at birth. So this therapy has already been approved by FDA and is commercially available. When we talk about this carrier, there are a lot of different options available. So the carrier we are using is called AAV9, which you will be hearing about again and again. And this is a carrier that shows good efficiency in skeletal muscle with the least amount of symptoms. So we started with our work on this gene therapy. And Gustavo has already talked about the whole process of developing a therapy and moving into the clinic. So I'm not going to go into a lot of details. But what I'm going to say is that the first and the most crucial need that we had when we started this work was a need of disease model. And when we think about a disease model, we have to find a system that can copy the human disease as closely as possible, because we have to look for how effective the therapy is. Is it going in the specific organ? Do we see any toxicity in other organs? So we want to have, and it's a model showing all the symptoms of the severity, pathogenicity that we see in the patients in this model. And it is very important to have the disease model to have a successful therapy in the end. And once we have the disease model, we design a therapy and eventually test that therapy in the animal model, so which is the preclinical phase. And once we see any improvement in the animal model, we move to the next stage of safety as well as move to the clinic. So at this point, I want to give a big shout out to AFBS and the NM families, because when we started this work a few years back, we were here. We didn't have any animal model. We did not have any potential therapy. So we first started creating animal model with the support from AFBS. And we created a mouse model in which we deleted the 𝗘𝗪40 gene, very similar to patients. These mice do not make any 𝗘𝗪40 protein. So these are the normal mice, and these black bands are the normal 𝗘𝗪40 protein, and mutants don't make any protein. And like 𝗘𝗪40 patients, in mice also the disease is severe. Most of the mice with 𝗘𝗪40 mutations die within 1 to 2 weeks after birth. And these are the images, like these mice are normal, almost to their normal siblings at birth. But as the disease progresses, we see they become really weak and they show a failure to thrive. So because these mice are so young, we cannot do a lot of studies in them. But one of the easiest assay we can do on them is we can put this mice on their side and see how fast they can get up, just to get an idea how weak their muscle are. And let me show you an example. These are the control mice. We put them on side and they get up pretty fast on all their 4 feet and come back in a normal position. And now this is a mutant mice. We put on the side and it takes them a really long time to get back in a normal position, showing that they have overall muscle weakness, which we have quantified here, that amount of time that takes for the mutant mice to get up. So with this data, we are able to see that, okay, we mice have very severe form of disease like patients, and they have extreme muscle weakness. So this is potentially a good model for us to the next step to develop a therapy. So this is what we have done in the next step. We packaged our KLHL working copy of the gene into a AAV adeno-associated virus carrier, and that we injected in the mice right after birth. And then we monitor these mice over how long they live. So I'm going to show you some data from these studies. So these are the mice on top left, you see, which are untreated mice. So most of these mice die within a few days after birth. And on the right are the mice. These are the mutant mice, which would have been like this, But these are the mice that are treated with adeno-associated virus that carry a normal copy of the gene. And currently, our oldest mice are about 8 months old. So as you can see here, they look very similar to the control sibling. And this is the data from the survival where the oldest mice, blue line is the control mice, and the orange line are the treated mice. And this is the mutant mice. They all crash within first few weeks after birth. So this is very exciting for us. The lifespan in mice is about 2 to 3 years. So we are going to keep this mice and see how long they can survive. And this is the data from weight gain after the treatment. Initially after the treatment, these are the control mice. And initially after the treatment in mutants, the weight gain is slowly, but they eventually catch up. And we have quantified here that the mice treated with the therapy, they gain about 70 to 80% of the body weight compared to their control siblings. So we also look at not only weight gain and survival, but also the muscle function. And one of the simplest thing we do is we leave them in their cage and make videos that we can convert in these kind of diagram to see how far these mice can go and how well they're doing between 2 points from here to there. So blue is an example of the wild type mice or the normal mice, and orange is the mice that was treated with the gene therapy. We do not have any mice without the therapy because, just to remind you, they die really early on without therapy. And this is what we see 30 days after the therapy. By 1 week of age, these mice without any therapy do not move at all. So when we give the therapy, within 30 days, we gain about 50% of their normal activity. And by 3 months of age after the therapy, we see no differences in the distance or the time they move in their cages. And this is a video of, there's one mouse in this cage who is wild type mouse, normal mice, and one mouse which was mutant, and we provided the therapy. These mice are 8 months old right now. And it is even really hard for us to tell which one was a normal mouse, which one was a treated mice. But if you see, we put ear tags on this mice so we can read the number and tell them apart. But after 3 months of age, we stop seeing any differences in the treated mice compared to the controls. They are perfectly fine, normal, move around. So in addition to the activity, we also have gone back and seen, does the therapy affect the muscle fiber size? These patients I showed in the first slide have a lot of protein aggregation and amylin bodies in the muscle. These are the untreated muscle when they were day 6 old, and compared to the control mice, they already have very small muscle fibers here. But the treated mice show the size of muscle fibers, which is very similar to the control mice. And this is a fiber size distribution. So in a muscle, not every fiber is of the similar size. We see a range of muscle fiber size. So compared to the control mice, the mutant mice also get all their muscle fiber size distribution almost back to the normal. And on the right is the electron microscopy image so that we can go really deep down and look at the each actin myosin filament in the skeletal muscle. So by P3 or day 3 after birth, we start to see these amylin bodies within the muscle of the mutant mice. And at the bottom are the treated mice. These are the control mice with really nicely organized actin and myosin filament. And on the right are the mice that were treated with 𝗔 𝗙𝗂𝗂𝗏 virus. And again, we see that the muscle structures is stored in this mice. We do not see any protein aggregates that are seen in the mutant mice otherwise. In addition to the fiber size, we also look at are these muscle healthy enough to produce same amount of force compared to untreated mice or control mice. And on the left is an example where we stimulate the muscle with a range of different frequency. And we see both control and the mutant show similar amount of force production, suggesting that not only we are able to rescue the weight, we are able to rescue the muscle size, but as well as this muscle are functional and are able to produce a similar amount of force as a normal mice. So these are really exciting results in terms of efficacy or effectiveness of the therapy. The second component of any therapy is the safety. And now we are focusing more on the safety part. One of the things is 𝗙 𝗙𝗂𝗫 is skeletal muscle specific protein. So we have looked on the top, you see all this band. What we have done is we have looked at the 𝗘𝗪40 protein in all the different organs in the body. And first 4 bands are from different types of skeletal muscle here. And we see some expression in, a little bit of expression in heart as well, but we see there's no 𝗘𝗪40 protein in liver, kidney, brain, or lung, showing it's mostly muscle-specific protein. And then what we have done is we have looked at the mice that were treated with the virus. This mice did not have any 𝗘𝗪40 protein, and now they are making 𝗘𝗪40 new protein. And this protein is also present only in the muscle and to some extent in heart. And no protein expression is seen in the liver, kidney, brain, or lung. So that shows that the protein that is being made is expressed at the right organ and places in the body. And at the bottom, this colorful picture, this is from skeletal muscle. And we have stained that with 𝗘𝗪40 antibody, which is green in color. So this is a control muscle where we see that the 𝗘𝗪40 protein is present in the skeletal muscle and certain muscle fiber. And on the right is a treated muscle where we see very similar organization of the 𝗘𝗪40 protein in the treated muscle. So this shows us that the 𝗘𝗪40 protein is going in the right tissue in the body, and within the right tissue, it is expressing in the right location within the muscle. We have also looked at pathology of all the tissue in the body to see whether can we see any abnormal structures in other organs, because there are a number of gene therapies right now in the clinical trial. In some of the therapies, there have been toxicity event or adverse events associated with liver or some other organs. So we want to be really careful that we focus on all the potential toxicities that can happen, at least in the mouse model that we can see. So we have scanned, these are the control. On the left is a control panel with all the different organs. And right, these are the mice that were treated with the virus, 𝗘𝗪40 and 𝗪9. We do not see any differences in any of the organs between treated mice as well as the control mice. So we have looked at liver, kidney, lung, spleen, heart. We also have data from brain that I have not put here yet. So this gives us some kind of assurance that not only this therapy is effective, but so far we have not seen any toxicity at the doses that we are testing. So just to summarize this work, we have a disease model that faithfully models severe 𝗘𝗪40 caused Nemelin myopathy in terms of lifespan, muscle pathology, as well as muscle function. 𝗘𝗪40 gene replacement therapy during disease state can restore muscle growth, structure, and survival, which establish preclinical proof of concept for future clinical translation. And as we move towards clinic, we of course have to consider a number of factors that can happen from the translation from animal models to humans, which is an immune response. There could be some unknown toxicities, patient selection, age, disease severity. So we are already almost finished with first few steps of this therapy, and now we are moving to the safety and the clinical translation of the therapy. With that, I just want to thank all the members of my lab who have contributed to this work, especially Grace and Joanna, as well as members from the Beck's lab who have helped us with some of this work. Finally, thanks to all the foundations, AFBS and many other agencies that have funded us over the years, as well as all the families that have been part of this work. Thank you so much. And I'm happy to take any questions at this point.
Wow. Thank you so much, Dr. Gupta. Amazing work. Anybody have any questions? Okay, I do see a question in the chat. Do we have any understanding of the impact of larger presence of KLHL40 in heart muscle?
That's an excellent question. We have looked at the heart. I have not presented the data here. So we have collaborated with Seidman Lab that has extensively done a lot of work on heart in human patients, and they have analyzed the heart of normal as well as 𝗘𝗪40 knockout mice, and they did not see any abnormality with all the assays they have done. So right now we have no proof that it could be affecting heart function. Having said that, because this mice live only for 1 or 2 weeks, we still advise all our patients to, whenever they go for their doctor's checkup, to always get the heart function tested as well, just to make sure there's nothing shows up later.
Great. And it looks like Stacy shared some information in the chat. Stacy, do you want me to, I can read this, or let's see. Thank you so much, Dr. Gupta, for sharing these results. Your findings remind me of an early stages of therapeutic development of X-linked myotubular myopathy nearly 2 decades ago in mouse models of that disease. Those findings were then validated in a larger mammal model, providing additional evidence of safety, efficacy, paving the way for clinical trials in children 9 years ago. Given the encouraging results you've presented today, could you discuss how you will advance this therapy toward human testing?
Thank you for this question, Stacy. We are right now working with a number of manufacturing, regulatory, and clinical bodies to define the next step of this process. As Gus has already described the process very much in the detail, we are following all the necessary steps to make sure that we can take this therapy to the clinic as fast as we can.
Great, thank you for that. And then 𝗙𝗪𝘂, who is also AFBS's board president, has shared in the chat that her daughter has 𝗘LHL40 gene mutation, and she's organizing a group for those affected individuals to be informed on the next steps of this study. So please reach out to 𝗙𝗪𝘂. She's obviously on this webinar today, so you can connect with her here, or we're happy to connect you to her so you can be a part of that group. Oh, and there's her email right there. Okay, great. Any other comments or questions? Yeah. Lynn, go ahead.
I just want to say I'm Lynn Byer. I work on the AFBS board. I'm a volunteer board member for over, I guess, almost a decade now, which is crazy. Maybe a decade. I lose track. And my daughter is Greta, who's 18 with Nemaline myopathy, and she had an opportunity to spend a couple weeks in Dr. Gupta's lab last summer doing research. She's very interested in getting involved in research, and I was there on the side, Secret Service, just helping with her care if needed. And I just have to say it was a beautiful experience. And her lab is just so well organized, and the people that work with her lab, Grace and all of the lab assistants, were just fantastic. So it was a wonderful opportunity for Greta and myself to see the back end of what's happening, and it's just really exciting work. So we're just thrilled to have Dr. Gupta continuing to work with the foundation and our community. She's a gem. So just want to say that publicly.
Thanks, Lynn. The bench is still available for you and Greta to come back at any time.
Thank you.
Love that. Well, Dr. Gupta, we're so grateful for the work that you're doing and for taking the time to be here with us today. So thank you so much. And now, if there's no other questions, I'm going to pass it over to Dr. Dowling, who will be sharing an update on his latest work.
Can everyone see that and hear me
Yes.
okay?
Yes, thank you.
So first, I want to apologize for being a little bit late, and also want to thank Vandana for stepping up and changing places. I can tell people why later, but I'm sorry for having been late, and thank you for welcoming me and inviting me to speak today. I'm going to be focusing specifically on work related to nebulin-related Nemaline myopathy. Though I think some of our ways that we're thinking about therapy development certainly have a lot of applicability across different genetic subtypes of Nemaline myopathy. And we're also starting to get more interested in things like gene editing, specifically for things like the ACTA1 gene. So stay tuned for that in the future. I don't think so. Just to lay the groundwork, the work I'm going to be talking about has been a really amazing collaboration between two other laboratories that probably people might be familiar with, Henk Grenzier's lab and Koen Ottenheim's lab. It's been a really beautiful partnership between the three groups, and I'm very inspired by them. They're great scientists and people, so it's been really fun to work with them. We've also looped in Hicham Taswoot, who's at the University of Washington, into this work and gotten him excited about Nebulin. So that's been a nice thing as well. And I just want to say that the work has been funded in part by NIH in the past, and we're trying to get more NIH funding now, some generous donor support, and from AFBS. And I should also mention that many years ago, we had AFBS funding to establish the first zebrafish model of NEB-related NEM1 myopathy, and we continue to use that model today. So that's been an exciting thing to have. I don't think I need to describe to this group about Nebulin, so I'm just going to skip. I think everyone is familiar with Nebulin being a common cause of NEM1 myopathy. In fact, the most common genetic cause of NEM1 myopathy, that 50% of individuals with Nebulin have mutations in Nebulin. This is a recessive condition, so individuals have to have a mutation on each of their copies of Nebulin. Right now, as is the case really across the entire NEM1 myopathy spectrum, there are no specific therapies related to Nebulin for NEM1 myopathy. One of the interesting things, and this is now motivating us to think about therapy in a new way, is the fact that Nebulin is a gigantic gene. It's in fact the second largest coding sequence in the entire human genome. Only Titan is larger. It was originally named because it was so big and no one could figure out what it did because it was so big. So it was called Nebulin because it was a nebulous function. But now, thanks to the work in part by Henk Grenzier, we know quite a bit about what it does. But one of the mysteries has been, why is it that very small mutations within Nebulin cause the whole protein not to work right? Many of the mutations, in fact, the most common one, the exon 55 deletion, which is probably the most common NEM1 myopathy mutation, removes only a tiny little bit of the protein. So it removes 33 amino acids out of about, see if I do the math right, like almost 10,000 amino acids. So it was mysterious to us why removing such a tiny part of it caused the whole protein not to work right. So part of the project has been motivating by trying to figure this out. And we turned again to the zebrafish as a way to try to model this. And the beauty of the zebrafish is we can actually make many mutations very easily. So what we've been doing is systematically removing parts of the Nebulin sequence to see if we can recapitulate what happens in patients and understand why it's happening. And I'm going to point out a little bit of protein structure, so forgive me for this. So Nebulin has unique N, so it's N-terminus and C-terminus. The beginning and end parts are unique. The N-terminus is what caps at the end of the actin thin filament, and the C-terminus is what lodges it into what's called the Z-band and anchors it, and that's how it provides its structural role. And then Nebulin is there to enable the actin thin filament to then wrap around it to establish its length and also to generate force. Most of the protein is composed of this big middle part that has these things called super repeats. And the super repeats are made of simple repeats, and you get 7 simple repeats in a row, and then you get a super repeat. And these occur, there's 27 of them in humans, there's 21 in zebrafish. And these small little deletions like the exon 55 deletion occurs within the super repeat domain, and in fact removes one little part of a simple repeat. And so the hypothesis we had is that the reason why those little mutations cause disease is that they make the protein not have the right conformation because it interrupts the super repeat. And that's what causes it to go away and become unstable and not work properly. And based on that, we thought, well, if we can expand the mutation and make it the entire length of a super repeat and remove it all, that should actually remove the problem, keep the protein stable, a little bit smaller, but stable, and still have function. And the way that we went about studying that is we made a whole bunch of different zebrafish deletions, either of one little piece of a super repeat or of full super repeats. And what we found is that if we remove any one little piece of a super repeat, so one exon or one part, one simple repeat, that causes the protein to become unstable and causes basically the full Nebulin phenotype that we see that would be the same as if we knocked the whole gene out. And it causes the fish to have disorganized muscle, to not generate enough force, to not swim well, and to also have reduced survival. So similar to the other models of Nebulin that we've made. And importantly then, when we remove the whole super repeat, we actually get no problems. The fish they look normal, their muscles are organized properly, they generate enough force, they swim normally, and they also don't have a change in survival. So this basically showed us that we were correct in our hypothesis that it was somehow by removing just a little piece that caused the protein to not function properly. But we wanted to really understand why that was. And what we also noticed is that when you remove a little piece, you actually lose the expression of nebulin. So it's probably best shown here. This is what's called an immunostain. These are muscle fibers that we take from the fish and we can grow them on a little plate. And what's really cool is they look basically just like human muscle fibers, and we can do a lot of different things to study the fibers once we isolate them. And this is what basically looking at the expression of the nebulin protein. And you can see here when we remove just one little piece of it, we get the protein goes away. So it should be here in green, and you see no green. But when we remove that whole super repeat, we actually get a normal green stain. So basically, remove a small piece, you lose the protein, remove the bigger piece, everything seems to be fine. The reason why this occurs is because when you remove only a small piece, the protein becomes unstable. It gets turned over too quickly. And we did a pretty clever trick to figure this out. We introduced into the nebulin gene in the zebrafish a green marker called dendra. And this green marker is really very cool because if you shine a light on it, it turns into red. And every new nebulin now will be red, and you can, based on the red to green conversion, you can measure the half-life of the protein. So you can figure out how long it sticks around. Nebulin normally sticks around for weeks. It's one of the most stable proteins in our whole proteome. But when we make these teeny little mutations, it causes the protein to turn over on the order of a day or two. And so here's the turnover kinetics, and you can see when we remove the whole super repeat, we get no change in the kinetics, and it goes on for days and days and days. You can see the half-life here, almost 50 days. And here now the half-life on the order of like 3 or 4 days. And that's the experiment. This was done again in 𝗏. He kind of figured out how to do this. This is pretty incredible that we were able to figure out how to do this. So this is just to summarize everything. So basically, if we remove one part of a super repeat, we cause the protein to be unstable and to go away, and then the fish not to generate normal force, not have normal swimming, and also die early. If we remove a whole super repeat, everything seems to be fine. We've now shown that we can actually remove several super repeats. In this case, we removed 5 of them, and when we remove 5 of them, the fish is still totally fine and makes a normal amount of protein. And this argues that nebulin can actually be created a bit smaller, so we can miniaturize nebulin by removing parts of it as long as we maintain that super repeat structure, as long as we don't interrupt the repeat structure. Of course, you can't remove too much of it. And if here we removed 18 super repeats, and now we lose the protein expression, and the fish behave more like a nebulin model of the mutation. So you can remove some. We're now trying to figure out how much. So basically, what we're doing is we're making bigger and bigger and bigger deletions until we get to the point that the fish are no longer having normal protein expression and having normal behavior. And through this, we'll be able to figure out what the minimum requirement of size is for nebulin. So just to summarize, remove a small little piece of the super repeat, the protein becomes unstable. And this is what most of the mutations are. So most patients have mutations that remove small parts of the super repeat, and it causes the protein to be unstable and lead to reduced amounts of nebulin, and that's what causes the disease. Remove a full super repeat, and you can prevent that problem and have a stable protein, and at least in zebrafish and also in mice, no evidence of disease. So this led us to hypothesize a potential therapeutic strategy. And this therapeutic strategy is, so what if we took the situation when there's one deletion that removes a small piece and made it bigger to remove a whole super repeat? Would that actually be effective in restoring protein expression? And so the way we've tried to do this is using a pretty old technology, or now becoming old technology, of using antisense oligonucleotides to do exon skipping. So this is something that's been done in the Duchenne field for many years, and also in spinal muscular atrophy was the first approved antisense oligonucleotide for exon skipping. And what we've been able to do is develop these antisense oligonucleotides to skip different exons, both in the mouse gene and also the human gene, to see if we can create a combination of them that turns small deletions into full super repeat deletions, which should hopefully make a stable protein based on our zebrafish work. And so we've been able to do this. We've created these different ASO combinations. We can show that we can get a deletion that is the full super repeat deletion. I'll skip forward. Well, you can't I skip all the way to here, because when we do that, we can show in mouse cells that we can get the full exon skipping, and also in human cells. And then we tested it in our mouse model of nebulin-related Nemelin myopathy. And what we can show is that we basically could, when we inject it just into the muscle, we can actually get protein expression restored. But when we try to do it in the whole animal, it doesn't really work. And so this is just showing this approach. So these are the morpholinos to do the exon skipping. No skipping, you are missing a part of the super repeat. It's not intact. The protein gets degraded. Full skipping, you get an intact super repeat. And so when we do this in the nebulin exon 55 mice, which have just the small little piece missing, without the injections, we see a loss of the nebulin protein or a reduction of it. When we add in the morpholinos and skip a full super repeat, we now get restoration of protein expression pretty impressively. So again, the problem with this approach is that we can do it by just injecting it right into the muscle. But if we give it to the whole animal, we don't seem to get enough of the exon skipping in order to restore the protein expression. So this is where we are now. We're starting to look at new generations of morpholinos or antisense oligonucleotides to get better exon skipping. We're also trying this new strategy of putting them all together with little linkers. So it's basically one super drug. And then the other thing that we're doing in parallel is we're trying to develop a different way of delivering antisense oligonucleotides using something called lipid nanoparticles. And that's a whole story for a different time. And I'm happy to field questions about nanoparticles and their potential role for therapy. So this is something that we're involved in. We're also thinking about some alternative strategies, such as gene editing as a way to remove the whole thing. But this is something we're very actively working on. I think I'm doing okay on time. All right. So the other thing I showed, right, is that we could remove more and more of these super repeats to make smaller and smaller nebulins. So the hypothesis that comes from that is that potentially you could make a gene therapy of a miniaturized nebulin, and that should hopefully have function. Because the problem with nebulin, unlike 𝗪HL40, which is of a size to fit into these gene therapy vectors, into the AAVs that Vandana was talking about, nebulin is way too big. It's in fact about 5 times too big to fit into an AAV. And we already have shown, based on our zebrafish work and some mouse work, that you can't make nebulin this small enough to fit into one AAV. So the question is, on the zebrafish side and the mouse side, we're trying to figure out how small we can make it. And now what we're also doing is we're making a different kind of gene therapy that basically stitches together proteins to make them bigger. This is a really cool approach called protein stitching or protein transplicing. And the basic idea is you can take a piece of a protein and add like a little part of a puzzle piece on it. And then you can take another piece of the protein and add another puzzle piece on it. Those two puzzle pieces find each other. They remove themselves and stitch the protein together. It's basically like you see these like little locked together. Once they lock together, they remove themselves and then they fuse the protein together. In this way, we can make miniaturized nebulins, and we can put them into multiple AAVs and then make basically a mix of them, like a cocktail of different AAVs, and then deliver them as one therapeutic. So we have now tried this for a 3-fragment miniaturized nebulin using protein transplicing. This creates a nebulin almost half the size of normal. So definitely not close to the full length, but definitely bigger than what we would get with just one fragment. So we were able to show, this is again the work of 𝗪𝘱 𝗪𝘂𝗪 at the University of Washington. He made these constructs. So he has done this for the dystrophin gene first. So he has a lot of experience on how to put these things together. And he was able to make a 3-fragment nebulin with these enteins, which are the little stitchy parts, and show that he can make a big nebulin right here. So this is the MIDI nebulin with the 3 fragments. we could then inject them into the mouse muscle and show that we can create in the mouse the same 3-piece fragment, so the big fragment. You also see that we get smaller fragments too. And we're not sure if the small fragments are a problem or not, but the important point here is that we can make this bigger fragment. So it's basically 3 times bigger than you'd get with using one AAV gene therapy. Interestingly, when we do this and we inject at around 8 weeks of age, we're able to extend significantly the survival of the Nebulin exon 55 mice that we have. We see some slight improvements in the pathology in the muscle, but we don't see much in terms of increasing strength or force generation. So it seems like it's a very mild rescue. And now what we're looking at is whether we can make more expressing of the 3 fragments, but then also try to make 4 fragments to make a bigger nebulin to see if that might be the solution. And so that's basically where we're at now. We're finishing this 3-fragment study, but we're changing it slightly to make better fragments and give more of it to see if that might be a solution. And then we're also making both 4 and 5 fragment miniaturized nebulin. And you can see it's going to be both like about 65% and about 80+%. And we think the 5 fragment for sure should rescue the phenotype, but it might be hard to make enough of that. So that's something, because no one's ever made something this large before. So this will be a new thing to try. So in the last little bit, I think I have a few more minutes. Yes, Jen? Okay. Yeah. I wanted to talk about a new approach that we're trying, and also reach out to see if patients or families are interested in contributing. So one of the challenges is with all of these therapies is we're doing a lot in mice. Can we use basically patient avatars or patient muscle to test these therapies as a way to see, A, if they'd work, but also with all of the, particularly with Nebulin and with ACTA1, there's many different mutations that cause the disease. So we're not certain necessarily that a therapy will work across every mutation for one of the genetic subtypes. So one of the ways we're trying to basically test this question is by making a big library of patient-derived muscle. And let me show you how that we're doing this. So this is a pretty emerging technique. And David Mack, who I think AFBS is also funding to make myotubes, taught us how to do this. So I want to make sure I give him credit. And a company that he's involved in called Curie Bioscience really helped us establish this. Some of our innovations relate to the fact that we've come up with a new way of making these things called induced pluripotent stem cells, or iPS cells. So we've come up with a way of making them very quickly, on the order of 4 to 6 weeks, and very cheaply. So this is one of the big barriers to using these kind of things right now. iPS cells typically cost about $15,000 to make for a specific mutation. We're able to now do this for individual patient lines for under $1,000. So it gives us the opportunity to make dozens to hundreds of them. And we've been able to show using these that we can turn them into myoblasts and then into myotubes, and that these myotubes, and this is what they look like. They're hanging off this little device. I think David had some hand in making this device, so it's very interesting. And this is basically a little muscle, and it will contract, and it looks just like muscle under the microscope. So we've done this for Duchenne muscular dystrophy myotubes because there's a lot of data on this to show that we can actually do the technique. And what you can see here, this is the force generation that, unsurprisingly, DMD myotubes have reduced force generation. Importantly, we've been able to show that we can give therapies to these cells. And so this is using a relatively new form of AAV called myoAAV4A, which gets to muscle way better than the AAV9. So Vandana will have to talk about AAV choice, because I think there's some really great data showing that these newer myoAAVs can be given at much lower doses and much less expensively, and also with hopefully fewer side effects. But our myotube system is really great for testing these. So basically, just to summarize real quickly, we developed a novel rapid protocol for making the iPS cells, which we can do it from patient blood. Basically, a blood sample is all we need, and we can make iPS cells. We can then turn them into muscle in a dish, starting from the patient blood cells. And we can use them to ask questions about pathology, about mechanisms of disease, and then also about therapeutic response. And so our goal is to develop a library of these cells from patients with all forms of NEM1 myopathy. And the hope is that we would both establish these to study in our own lab for the therapies we're making, but then also make it a resource that's available to the research community. Essentially, we're looking eventually to create kind of an international biobank of muscle diseases with these. So we're very interested in making sure that we do things that are sustainable and shareable. So if you're interested in participating, you can reach out to me. Here's my email. And I think I talked super fast. And so I've reached my conclusion. Obviously, these hands have not done an experiment in now more than like 15 years. And it's my amazing lab, first at SickKids and now at the University of Pennsylvania, that's done all this work. And also my amazing collaborators. And one of the great things about the NEM1 myopathy field is it's incredibly collaborative. I think AFBS should get a lot of credit for that collaboration. You really foster a spirit of interrelationship between the different researchers, scientists, and clinicians. And I think we all work together in the spirit of hopefully trying to develop meaningful therapies for patients with NEM1 myopathy. And I apologize for not having, and Jen, maybe you can send me the right logo so that I can have on my slide for AFBS. But yeah, so I will stop there and I will take any questions.
Thank you so much, Dr. Dowling. Amazing work, amazing presentation. We're so grateful to have you investing your time and effort into NEM1 myopathy. So, and thank you for being here. Anybody have any questions or comments for Dr. Dowling? A lot of applause. I'm seeing a lot of applause.
There's a lot of information, and I apologize for getting in the weeds a little bit with protein and stuff. But so it's not a muscle. So let's see, Lori asked a question. It's not actually a muscle donation. It's just a blood draw. So basically what we do is we get a regular tube of blood, and we then isolate the blood cells from that and turn them into these induced pluripotent stem cells. And once we have the stem cells, we can turn those into muscle, into heart, into brain. I mean, I'm in quotes, and we can take the cells that make them, and then we can make these organoid cultures that resemble the tissue in a patient.
I have a question for you. Hello.
Yes.
Thank you so much for your presentation. This was great. I know that your work has focused a lot on the exon 55 specific subtype of NEB-related NEM1 myopathy. Can you tell us how that work, which is really looked at a deletion that's really isolated to exon 55, how does your work apply to mutations, deletions, just across the whole nebulin gene?
Yeah, thanks, Stacy. So the goal is that these therapies we think would be applicable to a very broad range of patients with nebulin. One of the reasons we're motivated to make this library of patient-derived myotubes is because then we can actually test to see whether we're seeing therapeutic response across different mutations. So our goal is eventually to make sets of these, if we can succeed in making these multi-exon skippers, to make them across the whole gene, because you can imagine basically any mutation within a super repeat, we could skip to a full super repeat and it should correct them or ameliorate them. The miniaturized nebulin should be applicable to anyone with nebulin-related NEM1 myopathy. And then we're also looking at some strategies related to trying to globally increase the expression of nebulin in NEM1 myopathy. And we think that would be a very generally applicable strategy across the full mutational spectrum. But it is because we've focused on the exon 55, there is that caveat of question as to whether these things will be applicable. So we are making a few other mouse models as well. We've made, I think, a couple more mutations to see as a model to try to test different therapeutic approaches in different mutations in the mouse. And then I think we've got 3 or 4 different mutation lines that we're creating now in the myotubes And the hope would be to have dozens of those so that we could easily test our therapies to see, once we basically established what therapy looks like in these models, to say, yes, it would work or doesn't work in different mutation subtypes.
Okay, great. Thank you.
All right, I think we could take one more question, and then I know Dr. Darling.
Yeah, so Lori's asking about therapeutic timelines. This is always a really great and challenging question. Right now, we're at the point of we're testing, I think, 3 or 4 different therapeutic strategies all in parallel. None are yet ready to advance beyond the stage of sort of that discovery testing in mice. And once we have one that is, then we'll be able to start looking at what the late stage preclinical development pipeline looks like, in terms of where we would, what we need to do from a manufacturing perspective, how we're going to do the safety and toxicity studies, talk to FDA about whether we'll need to do large animal studies, what kind of toxicity package will be required. So that's will be dependent on the type of therapy, and is still a question I can't answer until we're a little further. We're very much hoping by sometime next year we'll have figured out that one of these is definitely our sort of lead one that we want to move forward. But we keep pushing on all of them, because at this point they all seem like they could work, and we want to make sure that we give the many different possibilities. Yes, we also have it's possible to make iPS cells of healthy muscle cells and bring them. Yeah, that's a great question. This has been something that the muscle disease field has been thinking about since the 80s. This idea of basically giving stem cells healthy stem cells to try to correct muscle disease. The challenge is that, so you can isolate stem cells in culture, you can make them. They don't survive very well, even in culture. And then even to give them to a mouse, which is, like the size of one of your muscles, in enough that you can see some meaningful rescue in that mouse, you need to give like millions of cells. And it's never proven to be, the muscle it's got a privileged barrier to it that really prevents cells from getting in. I've had a discussion about this sort of weirdly. You think about, as a tangent, you think about oncology and how, tumors metastasize to different places. The one place that tumors typically don't metastasize is muscle. And that's because muscle is really good at preventing cells from getting into it. And so that's, good from a oncogenesis perspective, but bad from a cell therapy perspective. So it's really hard. So we're actually thinking about, because there are a few tumors that can get into muscle, can we learn how they get into muscle and use those cues to then create better honing of therapies into muscle? Because obviously cells are big and they figured out a way to get into the muscle. So cell therapy really hasn't worked yet. I know there's still a lot of labs that are working on it, but I think a lot of us are still skeptical at the present.
If you have time for one more, I know Lynn has her hand raised.
Yeah.
It's okay. I can follow up later if he wants to take Beach's question in the chat.
Well, quickly, I can answer Beach's question. I think, the parameters for the first in human clinical trials will depend on the therapy, the indication. Some therapies are either better to try first in adults, potentially because of risk-benefit ratio, or because there's already some safety information in adults. Some are better to try in pediatrics. There's been a lot of examples in diseases that span that are lifespan encompassing, for example, like myotonic dystrophy, where trials have either been first in adults or first in humans or first in kids, depending on the nature of the condition and also the nature of the therapy. So hard to answer right now, but I think, there would obviously be an intention, if the therapy was applicable for adults, that adults would be included in hopefully getting it.
Dr. Darling, I'll just say one last thing. If you could just send contact information to Jen for how we can get blood samples to you, I'm sure we'll have a lot, my family included, that would absolutely jump on this. And then just information on whether we need to get you confirmation of the specific gene mutation, or if that's something your lab, I don't know how all of this works.
Yeah,
so
I can give that all to Jen.
Great.
Yeah, we'd love to share that and get this rolling. Absolutely.
All right. Yeah, amazing.
Okay. Thank you so much, Dr. Dowling. We're
Thanks,
so
everyone. I'm
grateful
going to
for
have
your
to jump
time.
off too. So
Okay.
it's great to see everyone today.
Yep.
Thank you.
All right. Thanks. Take care. All right. Well, wonderful to hear his work, and also from Dr. Gupta. Thank you both so much for taking the time to share this. It's really so helpful, I think, for all of us to hear what the latest is and what advancements are being made. I definitely still want to open it up to questions about anything related to the research presentations that you didn't get a chance to ask already, or now we want to open it up to any questions, just broader questions related to the information that Gus and I shared earlier in the presentation. And yes, Adrian, this is recorded, so we will share the full video presentation, and then we can also share we can certainly share our slides. I'll have to talk to Dr. Dowling since he had to drop off. And Dr. Gupta, I don't know if you want to, if you can share, if we can share your slides. Oh, you're on mute. We can't hear you.
Thanks, Jen. We are happy to share part of this work. Please tell us the important slides you would need. Also keeping in consideration, this is all unpublished data right now. New work from our group that we have not published right now. So sometimes it's difficult for us to have all the unpublished work in the public domain. But please email and we'll be happy to provide the best possible solution to your need.
Great. Okay, that makes sense. Thank you. Lynn, yeah.
Oops, I'm muting myself now. I just want to say, as a mom to an 18-year-old with the Nebulin mutation, and pretty severe, certainly with the trach ventilator, and someone who's worked on the board for many years, obviously there's so much exciting research happening, and research, as you can see, it does take time, and it takes time with all the different steps and getting to human trials. And, we're all very excited about this, but what everybody can do in the meantime is, if you haven't registered, with, the CMDR and things like that, like, make sure you do that. Like, if we get, if there's promising something happens and we have to go to pharma, like, we have to have the numbers. We have to show them who's in our community. And we cannot do that if people are not registered. And we don't want to get to a treatment and then not have people in our system. So please take it. It does take some time to do those things, but it's so incredibly important when we are finally able to get to human trials. And at that stage, we need to move. We cannot, at that point, be trying to get people into the CMDIR and these different citizen health and things like that. So absolutely take some time to get this done. I beg of you to do this.
Thank you, Lynn. Stacy, yeah.
I just wanted to say thank you to everyone who worked so hard to put all this together and share all this information, and kind of ask that maybe you all keep my son in your thoughts. Tuesday, he's having his major orthopedic surgery, both hips, both feet. We're anticipating a 6 to 8 week stay at DuPont, and I've coordinated with Stacy, and his muscle biopsy will be going to Begg's laboratory. And in just a very random coincidence, I met someone whose son had NM. She's never met another person who even knew about NM. And his sample was one of the first to be taken at Begg's laboratory. He passed away about 15 years ago and was 15 years old. His name's Brendan. And I know that his sample's there, and to just have met them so randomly, and it was a small world, but to run into someone based upon a Facebook marketplace pickup and learn that they had a family member with NM who's never met anybody who even knows about this was just kind of like the stars aligned.
Wow, that is rare and amazing.
Yeah.
And
yeah,
thanks for sharing, Stacy. And we'll definitely be thinking about Wyatt as he goes into surgery.
Yeah, thank you. I appreciate it.
I know there was another comment. Let me scroll up a little bit. I did share links to the CMDIR and to Citizen Health, as Lynn was emphasizing the importance of these. So these are easily accessible for anybody who hasn't already participated. But I see that Anna shared, does someone know any research or so for LMOD3 related Namelin myopathy NEM10? Gus, do you have any knowledge of?
Unfortunately, there is very little research on development therapies for that. It's still more in the basic science stage, trying to understand better the interaction of LMOD2 with the actin filament. But yeah, we monitor the literature and the research that is going on there, and because it's a recessive inherited condition, some of the other approaches that we are funding, for example, for 𝗪𝗓 or other recessive conditions, some of the approaches could be translatable to LMOD2, and maybe Vandana have some more comments about this.
Yeah, definitely. 𝗔 𝗙𝗂𝗂𝗁𝗂𝗃 at University of Arizona, who is a collaborator with Hank Grinzier, their lab has a LMOD3 mouse model they're working on. So I think it will be a good idea to reach out to them, and I'm happy to connect AFBS with him to see how far they are in understanding the disease model and developing therapy.
Thank you. Adrian, you have a question?
I have a comment. We just here for great developments on science, and we can see that some of the questions and some of the registers are based mainly in USA. I encourage to everyone who is an international patient, we can also help you to get tissue. We can also help you to get blood, no matter where you are. I have done it with my son. I know 𝗪𝗪𝐶𝑖 has done it for her daughter. So if you find issues just to, hey, I'm not in America, I'm not in mainland Europe, I'm in some parts of Asia or something like that, don't hesitate to contact us, and we can help you on make this happen, because at the end, the data, this is worldwide data, and for obvious reasons, we are collecting this in US and in Europe, but we don't want to just discourage someone who say, hey, I don't live in Boston, what can I do? Just let us know and we can help you on that. By the way, I also am in the board of AFBS.
Thank you so much, Adrian. Yeah, I just can't emphasize enough how much we want to be able to support the community and contributing to research. And so please always reach out to us with any questions and to help facilitate any of these contributions. 𝗪𝗂𝗂, yeah. Or was your hand, was that raised from previously? No. Okay. Let's see. I see a couple comments in here. We're new to Namelin. Our daughter got diagnosed at 1 month old. We've tried to get information on ACTA1. Do you have a database you recommend for us to get more information? I think I have a follow-up question, and maybe if you want to expand on what sort of information you're looking for, if it's research information or connecting with other families, we certainly want to be able to support you and the community and getting as much information as possible that's helpful. Research and connecting with other families. Yeah. So in terms of connecting with other families, I highly encourage everybody to join. There's a wonderful Facebook group that 𝗪𝗪𝗂𝗂𝗁𝗂𝗃 Cassette helps facilitate. And it is a robust group of congenital myopathies, including Namelin myopathy families and affected individuals, where it's a great resource for asking questions, meeting other families with the same gene mutation, and in the Namelin myopathy community. Thank you, Stacy, for putting that in the chat. So I really encourage you to join that if you haven't already. It's been a really wonderful resource for the community. So that's, I would say, the first place to start. And we're also happy to make individual connections. So if we can set up some time to talk and find out what you're looking for, and maybe we can make a personal connection with another family to ask questions in addition to that Facebook group. And then in terms of research, we have the latest in what AFBS is funding on our website. So if you go to the research page on our website, you can see the projects and also all published papers that are referencing any advancements in amyloid myopathy research that AFBS is funding. But I don't know if there's any other information that you want to share. Oh, and Gus, thank you. Just shared. It looks like a link to it. Go ahead, if you want to explain.
If you haven't seen that publication yet, this is a review published last year for the group in Australia that they were the researchers that discovered the first mutations in ACTA1 and link that mutation to the nemaline pathology. So you can have tons of references there, but you are always welcome to reach out to me or AFBS to ask for specific papers, or we have a collection of all the papers and we read it. Yeah, feel free to go to that link and read that paper, and you will get tons of citations about ACTA1.
Great. Thanks, Gus. And just to comment on some of the or add to the comments, 𝗨𝗪𝐶𝐵 mentioned that the AFBS's website and newsletter has great resources in addition to the care guidelines, which Stacy posted a link to. Really, really valuable resource in terms of care, especially for a newly diagnosed individual. And I'd also emphasize, I'm going to put in the chat here to join the AFBS's fill out the contact survey, and then you'll be added to, if you're not already on our email list, you'll be added to our email list. So we share resources regularly for both to the NM community and to our broader network. But you'll also be subscribed to receive a series of emails that really comprehensively outlines all the resources that are available to the amyloid myopathy community in a pace that's hopefully easy to digest while you're newly absorbing all the information on care and research. So I really encourage you to join through that, and you'll automatically be subscribed to that email series. Great for newly diagnosed families, but also for anyone, I think, that is just looking for a reminder on care and how to contribute to research. And all of this information is also on AFBS's website, which is buildingstrength.org. So I really encourage you to explore that website and also reach out to any of us. There's quite a few board members from AFBS on the call who are all amazing and wonderful resources. And Gus and I are also always available to support any of your questions. Did I miss any other comments or questions, or is there anything else that anybody would like to ask now while you have us live on the webinar? No? Okay. Well, thank you so much. And I just want to emphasize again how grateful. Oh, wait, Gus.
Sorry, yeah, I see one. I think we missed this one. Someone, Anna Schwartz, based in Europe with TPM2, newly diagnosed. Can I also register for the US database? Yes, of course, CMDIR. You can access to the website. I will put the website here. CMDIR.org, and yes, this is international registry, so everybody can.
Yes, please do. That would be great. It is, like I said, it's an international registry. This is not just US based, and so we have a global participation, and it is really very critical for everybody with NM to register, so we have accurate representation and understanding of Nemaline. Because it is a rare disease, it's really important for every single person to be contributing to our registries, both the CMDIR and our newer Citizen Health. Okay, let me just double check that there isn't anything else. Okay, great. Well, as always, please feel free to reach out to any of us. My email is on our website and also in all of our newsletters. So I'm always available, as is Gus and our team. So always please reach out. And thank you for taking the time to join today. For those of you who are able to make it, and for those hopefully watching this recording, I'm glad you're watching. Yep, just again, thank you to Dr. Gupta and Dr. Dowling for taking the time to share your work. We know that you're spending so much time and effort to make progress for Nemelin myopathy research, and we are just forever grateful for your time and energy towards that. So thank you again, and appreciate everybody being a part of this community and the work that we're doing. And I hope you have a great rest of your day.
Thank you, Jen.